Hello, welcome to the Incubator, the Biology Jobology podcast. Today we'll be joined by Dr. Jen Guarino, who did her PhD at Yale and is an Associate Medical Director at BGB Group. So Jen, what do you do now? What's a typical day?
So great question. I work for a medical writing agency called BGB Group, and essentially we are responsible for, our main goal is medical communication. So we partner with a pharmaceutical company, and we develop educational resources and materials for healthcare providers, educating on either disease states or on clinical trial data or clinical trial study designs that are ongoing to get feedback and information. So it's really varied. The projects that I work on can be extremely different. Can be anything from, as I mentioned, getting feedback from a thought leader or KOL on a study design and whether they think that it's asking the right questions and whether they think that it's addressing an unmet need or whether it's kind of overpopulating a market or things to that effect. Then it could be all the way to something that might be what you would call unbranded, which would be like disease state education or a therapeutic class education, so things that are not focused on development and bringing to market of a drug, but rather raising awareness in the cases of rare disease about like what the disease is and what the current therapeutic options are and what's being investigated, or as is the case in oncology now these days, educating on this new class of, immuno-oncology agents. So this can mean anything from putting together steering committee meetings or advisory boards, which are actual meetings where you get together with the thought leaders and you show them data or study designs and you ask them questions, and you get a lot of really valuable feedback. And like putting together meetings to something as material as a brochure that might be handed out at a conference or a slide deck that might be presented to a doctor, whether it's in their office or during a continuing medical education symposium or things like that. So the audience varies. It can be, again, what we call thought leaders or experts in the field. It could be community oncologists. It could be nurses. It could be sometimes even payers, so insurance folks or policymakers. I mean, the audience varies and the medium for which we produce the deliverable varies. So it's really interesting from that perspective, I would say.
Great. So how did you end up at BGB?
I actually started out working another agency doing publications work, and I actually realized that, and publications is, exactly how it sounds, even from grad school, you're really just writing papers on data, except that you have no control over the data or the story flow or anything. So, and it was interesting, and it was actually probably a good transition from grad school because I was very familiar, obviously, with writing papers, but it didn't, it felt a little bit too disconnected for me, and I wanted to be more involved in the actual communication of science, if you will. I know that obviously ACPs read papers, but you don't get to interact with them quite as much, and plus I wasn't really working in a disease state that was interesting to me, and so I started looking into other avenues for medcoms, and I kind of realized that there's all different kinds, and I didn't know that. I almost thought like pubs was it, and that's definitely not true.
So what other types are there?
Well, you can, like I said, basically all I was doing was writing papers, and now all of my projects vary. I could be putting together slide decks, I could be putting together FAQ documents, I could be putting together, well, a lot of my job is slide decks, but it depends on the end experience, whether it's for the ad boards or whether it's for MSLs or the sales force or policymakers or whatever. So it's a lot more varied in terms of the projects that I do. And it's not standard writing anymore. Now it's, you have to figure out how to convey information visually in large part, which can sometimes be challenging.
During your PhD, how did you prepare yourself for this job?
I think early on in my PhD, I realized that I didn't really want to do research. And so I really started investigating other avenues or other areas that I could use science and even hopefully use my PhD that I wasn't so minutely focused on a very specific topic like you are when you do research. And so I guess I started looking into I obviously looked into what other kinds of careers there were, and I started looking into things that I could do on campus that were outside of the lab. So I joined the Yale Journal of Biology and Medicine, and I worked as an editor there, which was very different and an interesting and unique experience. And I think that definitely helped me realize that I was much more interested in the communication of science than the actual doing of science, if you will. And then I got involved with the Biomedical Careers Committee, which was a small group of students at the time, yourself included. And we realized that there wasn't a lot of resources available on campus for career development and exploring careers that are outside of academia. And so we wanted to start essentially creating resources for other students and postdocs. And so we started this annual biomedical career fair, and it's still running today. And I think it's something that you and I are probably both very proud of. And I think that I use those outside experiences to help me leverage my translational skills into moving outside of academia. So that I wasn't just able to pipette, clear liquid from one test tube into the next, but that I had all these translatable skills that I had already started building and utilizing with these outside experiences.
What's been the biggest surprise for you as you've transitioned from moving clear liquids from one tube to another to writing?
Sitting, actually. I say that with some jest, but honestly, that was one of the hardest things to get used to, is not being active all day. And I guess the second thing I would say is that it's very different, I think, working in a lab versus working in a corporate office type situation, because your hours and your time and what you're focused on in lab really vary and can be really busy or somewhat slow. And although that same ebb and flow exists in the corporate environment, there's that constant need to be mentally on. So I found it actually kind of more exhausting at the end of the day. I felt like I was working much, not to say working harder, because I think that, everybody works hard in grad school, if only to get out of it. But I just think that there was like a different level of focus. So I think that was very different. And then the final, I guess one other thing that I think is very different, and I alluded to earlier, is that you don't have a lot of control over what you want to do or say. So you have a client, so they, really drive what it is that you need to be conveying, in whether it's a presentation or brochure or whatever. And you really have to stick to that. And there's a lot of strategy involved that there really isn't involved in academic research, because, and at the end of the day in academic research, you're just asking a question and answering the question. But in the corporate pharmaceutical world, you really need to make sure that your data is being interpreted correctly, and that there's just a lot more strategy involved, which is actually very interesting.
Yeah, it's like there's a psychology to it.
Yeah.
Because there's an element of customer service. In my current position, I have to phrase things differently than I would as an academic scientist, because I have to stay within our messaging in a way that I guess softens blows sometimes. Yeah,
absolutely. That's exactly what I mean. I think it is fascinating, and it's also sometimes tricky. And then, I work on this one thing where we have a pretty detailed manuscript of information we're allowed to use, and then there was so much behind every word that we put into those sentences that sometimes it makes it hard to then put them into bullet points, say, or essentially repurpose the content, because, like you said, there's language that goes into it that is very purposeful, which is interesting.
So, in your job, is it mostly PhDs, or do you have a mixture of backgrounds?
Yeah, it runs the gamut, really. So in an agency, you typically have a medical team and you have an account team, and the account team are the people that interface directly with the clients. The medical team has some interaction with the clients as well, but the account team is the main sort of point of contact. And then we also have the luxury really of having an entire creative department. So we've got, I guess you don't throw editorial into that. We have an editorial department and then a creative department. So we have art team, we have a copy team, we have a digital team, and so forth. And so certainly not everyone has PhDs. Of our medical team, actually all of our medical team either has master's, PharmDs, or PhDs. Beyond that, just a bachelor's really. So it's a very, very wide range, I'd say.
How has that been for you? Have you enjoyed working with more creative types?
Good question. It's good and bad. It's good because I don't think like that. And so it's very useful to have someone who is creative and I can say, okay, well, this is the information we need to convey. And if it was me, I would be drawing a stick figure. So I could really use your help in, helping me make something that's more visually appealing, but still makes sense. So from that perspective, it's awesome. Sometimes there can be challenges, I think. And I think it's almost like a analytical thinker versus a creative thinker type thing. So the communication can sometimes, as funny as it is that we're a communications agency, communication can sometimes be challenging because they need different information than I need and vice versa, So, but it's very, I think in the long run, it's essential. And it's certainly good from a development standpoint.
Or at least from a humanistic point of view, where some people just have a different operating system than you do. And you have to deal with that, understand the things that really work for you may not necessarily work for them.
Yeah, 100%.
What's the most rewarding and exciting part of your position?
I think, I am lucky enough that I work in oncology. And I think that that's part of what I think is the most rewarding and exciting part because it's such a fast-paced field and research is constantly evolving and new drugs are being approved all the time. And it's just a really exciting space. And it's also an exciting era in oncology because with the advent of immuno-oncology, or I guess not the advent, but really the development, because for a long time we looked into immunotherapy, but there wasn't a lot of excitement behind it. And of late, we've discovered these classes of immuno-oncology drugs, and they're working in ways that we didn't understand and we didn't think possible. And I think it's that is so exciting. And you hear these, anecdotal patient stories, and it's very rewarding to know that in some capacity, albeit I'm not a medical doctor, I'm not treating patients, but I'm helping to educate physicians and I'm helping to get clinical trial data out there for people to see and understand and evaluate and turn into new important questions. And I just think that it's really, truly for me, it's the therapeutic area that excites me.
Oh, I think all that stuff with PD-1, PD-L1, and CTLA-4 is so exciting. It feels like every week I'm seeing a new article in Science or Cell explaining the new stuff that's popping up. And as you were speaking, I was thinking about how essential it is for patients and clinicians to understand the promise as well as the limitations of this, because it's not this miracle drug yet. There's still some cancers that are rather intractable to this treatment, and some develop resistance. What you were doing, your company's doing, is so essential to tell people it can do these amazing things, but there are limitations. We still don't know why it works for this subset of patients and what those markers are and so on. So it's really exciting. Yeah,
I think you hit it. Sorry to interrupt you, but I think you really hit the nail on the head. There's so much we don't know. And I think that that's one of the reasons that biomarkers are going to become such an important part of understanding this therapy. In the past, biomarkers have been very binary. Yes, you'll be, yes, this therapy will help you. No, it will not. Sort of. And now, we're going to need, I think, a whole range of biomarkers to really select a patient population that's going to benefit from one drug over another. And we're not there yet. And I absolutely agree with you that there are definitely limitations. And there's certainly, even though it doesn't have the same chemotherapy side effects. So it's not there yet, but it is exciting.
So currently, how are you developing yourself?
That's a good question. I think, I seek out opportunities to work on new things. So, in fact, I've had a lot of experience working commercially. And I think right now, a lot of the work I'm doing is a little bit more medically focused, which is different for me from a job perspective and from my experience in this job, which I like. And I'm also getting to work on a couple of like different types of projects. So an advisory board, for example, I'm getting to do an advisory board for essentially the second time, but more from the first time as like the lead content developer. So that'll be really exciting. And I think it's important to both seek out things that that you have never done before and grow in that way. But also keeping up with, the literature, like I said, we talked about it constantly every day. There's a new paper, keeping up with the competitors, understanding what trials they're initiating, what avenues they're investigating. So I think keeping up with the science of immuno-oncology is really important. And yeah, just advocating for yourself. And I now work with a medical writer, and so having responsibilities of someone else's development is a whole new area, and I think that's going to be a good challenge that I'm looking forward to.
So you mentioned that you're now working on an advisory board. How did you find that opportunity?
Well, it honestly has a little bit to do with supply and demand.
Oh, Jen, you've gone full corporate. You're speaking economics now.
Oh, yeah, I know. Well, there was a lot of work coming in on one of our teams, and our commercial work had subsided for a little while. And so I was talking to my supervisor and talking to some of the other people on the team, trying to figure out, how we can best help support this other team that's having this influx of work. And there was a few projects, and I basically said, this would be a cool project to work on because me and the person that I work with don't have any experience on it. And obviously we would need some oversight, but it would be a good opportunity and a learning experience for both of us. And they needed the help. And so that's, it worked out. But I think in general too, something that's similar to grad school, I guess, is that, I meet with my supervisor once every 2 weeks, and we go over the projects that we're working on, and we also go over development. So, and I talk to her, I say, hey, I'd really be interested in doing, X, Y, and Z. How can we make that happen? And for the most part, I don't do the resourcing, so that's up to her and up to the senior level management to decide who's doing what. So I just make it known that, this is a type of project that I've never worked on, and especially because I've had to change supervisor. So she's not necessarily familiar with what I used to work on, and I guess that's it.
How is your relationship with your supervisor different from your PI?
It more so your environment than the person. So I think at the end of the day, you're still responsible. That person is still potentially acting as a mentor for you, and you still have a responsibility to them to do a job and to get something accomplished. So I don't think that's different. I think that it's more than likely have a bit more flexibility in graduate school in terms of your day to day, what kinds of questions you're asking, what kinds of experiments you're running. Whereas in a more corporate environment, outside of the bench, you are a little bit more restricted in what you're doing, and not in a bad way, just in, you have specific deadlines, you have specific projects you need to complete. You still have flexibility in guiding your schedule to the degree in which you prioritize what you're doing, which I think is arguably quite similar. But I think it's that environment that makes the supervisor role somewhat different. So I hate to say that it's not collaborative in both environments, but I think it's probably more collaborative in a graduate school environment where you and your PI are working towards answering a question. And I just think that, like I said, there's just more flexibility, and you have a little bit more flexibility in what you do. Whereas in a corporate environment, you're still collaborating, you're still working together to solve a problem or to complete a project or whatever the case may be. But there is a lot more guardrails in terms of timing and in terms of responsibilities and things like that. I think that the roles themselves of a supervisor are very similar. It's just the environment that you are in is different. I guess there's also some level of well, I've had a couple of different supervisors actually at BGB, but my relationship with each of them has been distinct, but also I would argue maybe a little bit closer. I'm learning, I guess, I'm learning a lot. It feels to me like I'm learning a lot more from them, but I think what it is that I'm learning a lot of different types of things from them. So from your PI advisor, you're at the end, the end game is like very, you're very focused in what you're investigating, and there's only so much wiggle room that you might have in terms of what you're going to learn from your advisor. Whereas I think what I'm learning from my supervisors is it truly runs the gamut of everything from, and this is specific to my field, but how to stay on top of the literature, to how to interact with the client, to how to interact with a thought leader, to how to strategically position a drug in a really complicated market. Like, there's just so many different types of things that you're learning that I think kind of distinguishes that a little bit from the PI.
How's your work-life balance compared to grad school?
Pretty different in my case. I don't know if that would be true for everyone, because for me, my advisor in grad school was awesome, and he was, he seemed to be a proponent of a work-life balance, and he didn't expect, his students to be in the lab 24 hours a day, 7 days a week, making sure they're turning over research constantly. And I think that was really great, because it gave me the opportunity, especially in my case, to explore other careers and to get some experience doing things beyond the bench. So for me, my work-life balance is not quite as good now. I think we work theoretically 9 to 5:30, and those are set hours. And, if you're not going to be there, you have to take PTO day or you take a sick day or whatever it is. And I think that casualness of grad school, at least again in my position, is not there now. But I do think that I have a good work-life balance, especially in this current job. I think my, BGB is really great about making sure that when people request time off and take time off, that they're actually off and that they're not online all the time and reading emails and responding to emails. And there's really a structure for ensuring that there's always coverage. There's always someone to back you up, which I think is really great.
What's the most valuable skill you developed in grad school?
Honestly, I think I developed a lot of writing skills in graduate school. As much as it doesn't maybe seem like it, you do a lot of writing in grad school, whether it's for fake grant applications for the NSF that aren't fake, or writing your thesis, or writing your quals, or writing a paper. You do a lot of writing, and I think that was probably, the thing for me that probably progressed the most over grad school. I think I became much better at writing more succinctly and writing scientifically. And I think also in doing some of the extracurriculars, if you will, I learned not just how to write for science, but to write for different audiences, which is really important in this field.
What suggestions do you have for those interested in entering into the science communication field?
Get involved in things that aren't lab. Seek out any opportunity you can to be involved in communication in some capacity, whether it's writing a blog or joining a journal and learning the ins and outs of being an editor, or there's probably a unique experience to Yale. But just find things to do outside of lab that are related to communicating science. And I think that that's really important. And then also just get educated on the types of careers there are. I don't even know all the types of careers there are that you can do in this field. And I think that sometimes it's very difficult to find information about jobs outside of academia. If you don't have them, start a group, right, Will?
Yeah, clearly that's the best option.
Yeah.
Finally, what words of encouragement do you have for those who have failed their 100th Western blot or their 1,000th PCR?
Oh, feels like ages ago now. And I would just say that, we've all been there. Literally everyone that's gone to grad school has been there. And I think towards the middle of my PhD experience, I was really discouraged by quite a lot, by the failure of so many experiments and whatever. And I just think that you have to remember that there's a light at the end of the tunnel. It's not an indefinite experience. And at the same time, I think one of the most important things to keep in mind is that you are learning those transferable skills that you might not see, but are there. Whether it's giving journal club presentations, you're working on not only how to present, but also how to put a presentation together. And just recognizing that all of your work is not for naught. Even though you failed your 100th Western blot or whatever, you've still learned troubleshooting, if nothing else. You've probably mastered the art of troubleshooting. And yeah, I think that you just have to remember that there is an end in sight, and that the choice, you don't have to do this for the rest of your life. There are a lot of opportunities out there to use your PhD and to use science and to find something that works for you and is something that you can be, if at least not passionate about, but really interested and excited about.
Great. Well, thank you, Jen.
Yeah.
A big thanks again to Jen and to Protein Tech for sponsoring, and thank you for tuning in. Don't forget to subscribe and rate us on iTunes. See you next month.